Summer is for slime molds, and if you are out there right now and looking at damp wood logs, you are definitely finding some really interesting things to look at. And I wanted to help you out so you know what exactly are you looking at. So let's start with the basics. What are slime molds? So according to literature, they are single-celled eukaryotic organisms. So they have what we call a true nuclei, a neat little membrane that it's enclosing all their DNA into a single location. Does that mean they have a single nucleus? That's not what I said. What I said is that all the genetic material have a membrane around it. But different types of slime molds will have different numbers of nuclei from one to thousands, maybe millions, we don't know. But all are feeding in the forest floor, and as diverse as they can be, there be feeding in yeast, bacteria, fungal spores, or other protists. They have different modalities. Most of the slime molds that we are going to encounter are going to be colony forming. So they will live their lives as an amoeba, but then when the environment sends the message, they're going to form together what we see as the reproductive structures or the sporangium. In the greater DC area, we are talking about Washington, DC, Maryland, and Northern Virginia. There have been 100 documented species of Myxogastria. But in the entire world, we are closer to 1,000 species. And these are in a wide range of habitats, namely the forest floor, the soil, logs, anywhere around the ground. And as Valerie was mentioning already, they are not really true fungi. They are amoebas. I'm trying to go to the chat here. Great. So, if you've been paying attention to the news, you may also have seen that they have been known to be intelligent creatures, based on this style of finding food, and we'll talk a little bit about that, but towards the end of the presentation, if we look. In any case, I wanted to take a page from our membership director, and tell you a little bit of slime mold in the news. So in 2024, scientists found that some types of slime molds can be in your morels. So if you look right here, you have the paper that published that finding. These are Chinese morels, but who knows? Maybe Marylanders have not been paying attention. So the next time spring comes around, take a look at your morels, and you may find some dictyostelid slime molds trying to eat them too. And then the most, oops, I clicked on the link. The most interesting news that I found while preparing for this presentation was this article where a designer is working with lichens and slime molds to make wearable fabrics. So the lichen fabric was pretty round the mill because the fungus will make a very substantial fabric, but then it blew my mind that it was making slime fabric also. So the individual that was used for this endeavor was Physarum polycephalum. So what the researcher or the artist did was to make a mesh-like material and then put Physarum polycephalum inside of that. The growth of the slime mold was creating these very intricate patterns into the fabric, which was very enjoyable and very appropriate for design purposes. But then you have to keep in mind that it's a living creature that could be very delicate. So the conditions for growth for that particular project needed to be very specific temperature, moisture, and the food that was given to the organism to grow. Once the fabric was done, you weren't done feeding it. You had to continue to feed the organism in order to maintain the fabric. So, I mean, if you're that dedicated, more power to you. But I think there's more to be developed. There's more to be researched in order to develop a technique that would be more appealing for the rest of the world. Do you want to feed your dress? I don't. Anyway, and then this is a wonderful graphic that I stole from the slime mold identification and appreciation Facebook group. And there is a link here that if you click, you'll be able to go directly to the group. In subsequent slides, I will be touching on different articles and also the slime mold in the news. So at the end of this presentation, you'll have a slide with the QR code so you can download the presentation and get the links to enjoy your leisure. So looking at the anatomy of a slime mold, you have what we can see with our naked eye in the forest floor, specifically more often than not in very damp, wet logs. And those are going to be the sporocarps, or the capsule where most of the spores are being produced. That sporocarp is covered, like Valerie mentioned, by a peridium. That is a membrane that covers the most of the spores and the threads that connect the spores, which are called capillicium. Now, if you look at the base of that sporangium, you may have a little cup that holds it together, holds it up, and that cup, it's called a caliculus. It might have some ornamentation. Part of this is also used for identification purposes. So it's important to use the microscope in order to bring the slime molds into species. Looking inside of the sporocarp, you'll have the spores that are attached to threads that hold them together, and that is called the capillicium. And the capillicium also will have ornamentation that you can use to identify the species using the microscope. And then there are the spores that may have warts or may have other types of ornamentation that help with ID. Yes, some slime molds can be stinky too. If you want to ask questions during the presentation, please feel free to put them in the chat. I am able to read and answer it and incorporate in the presentation as well. So there are three types of slime molds that are most commonly known. And the biggest group and the group that we are mostly going to identify in the forest is the plasmodial slime molds. Those are known as myxomycetes, and those are most commonly found in the forest. They're going to be large multinucleate, and it's an amoeboid mass. So it's a slime blob. They don't have any cell wall divisions, so that blob is full with thousands of nuclei that are not organized in individual cells. When is in the vegetative mode, the plasmodium, it's going to make a pseudopodia. So if you remember your basic biology class, the membrane of the plasmodium, it's going to move and engulf or circle the particle that they want to eat and incorporate it like that into the rest of the cell. So it's extensions of the membrane that come like little hands that are going to circle around the particle. When there is no more food that they like, that is a signal from the environment that they have to start reproducing. And it's at that time that we as humans enjoy their morphology because they can be quite intricate and quite beautiful. And such is the case of the Metatrichia vesparium down here. And if you click in this picture, you'll be taken to where I got that picture from. Slightly smaller, but maybe you'll see them if you're lucky and using a very powerful hand lens, are the cellular slime molds. These belong to the group of the dictyostelids. They're going to be single-celled solitary amoeba. So instead of living as a plasmodium, these are going to be as single amoeba, but they will still feed by engulfing the bacteria that they want to eat. However, when starvation is it's what's happening, meaning there's no more food, then they're going to get that signal. It's time to reproduce, but they're going to make a multicellular slug. So they're going to try to go the plasmodial way. They're going to be smelling the environment to find each other. And by smelling the environment, I mean quorum sensing. Every single amoeba leaves a chemical trail that tells other amoebas where they are. Sort of, where is my stinky friend? I need to follow my nose to find them. And by quorum sensing, they're going to come together and then make the sporangium to reproduce. And then there is the curious case, the mysterious case of the protostelids. These are single-cell solitary organisms as well, but even the fruiting body is one to a few cells. So you really need to be looking under the microscope in order to be able to see these guys. So for the purposes of tonight, we're gonna focus on the on the slime molds that you are most likely to see, which is the case of the plasmodial slime molds. And not to be confused with the slime molds, but utterly beautiful and morphologically similar is the case of myxobacteria. These are gram-negative bacteria that also live in the soil. You might confuse them with the dictyostelids, but these are not dictyostelids. These are protists, and they have no nuclei. These guys live as free bacteria in the soil, and very much like the slime molds, once they are starving, they're going to aggregate to produce a fruiting body. This fruiting body is indeed multicellular, and these at the top are called myxospores. They are resistant, meaning that the cell walls are going to be thicker. They're going to be able to resist desiccation. They are just little pockets of survival in order for the organism to wait until favorable conditions come again. So like I said, these guys do not have a nuclei, so these are not to be confused with the slime molds. But since they are so pretty, and if you're looking that close, you might be able to see some, I wanted to mention them here too. So the life cycle, I'm not going to get into too much detail since Valerie already mentioned these to you. Just around the life cycle, you have the spores. The spores germinate depending on the amount of water in the soil. They can go amoeba or they can go flagellated cell, which means there's a lot of water. I want to swim long distances. I need a motor behind me that I can circulate and move faster. So if I'm just in the soil and it is damp, but not really a whole lot of water, I might go amoeba. But if I am germinated in a soil that is saturated, then I'm going to get a signal to get a flagellum. In any case, when these germinate, they will continue divisions in the soil until it is time to reproduce. They're going to combine or get cytogamy, then karyogamy, which means they're combining the nuclei, and then they're gonna continue into the life cycle until they get the plasmodium. The plasmodium detects that there is no more food, at which point it's gonna start making the sporangium, and ta-da, the cycle starts again. One curious thing that happens when we compare these, oh Jesus, I clicked on the link again. This is how easy you're gonna have access to these papers, my people. In any case, when you compare that to the dictyostelid life cycle, you have something very similar, but in this case, the amoeba form an aggregation. There is no plasmodium phase. It's just going to produce an aggregate of individual cells. And here is the kicker or the funny part. If you arrive on time to the congregation, you might get to reproduce. But if you arrive late, you will just be peridium, or you will just be stalk. The speed at which you come back to your friends determines the position in the aggregate, which determines are you reproducing or are you just support. So if you're addictive, sell it, you better arrive early. Ay, Dios mío. Okay. So, what are some of the characteristics that you need to keep in mind in order to identify these fantastic creatures? At the macroscopic level, I would suggest bringing a hand lens. And with a hand lens, you can also put it behind your phone and make that a macro lens so you can take better pictures. So, what we're going to be looking at are some of the characteristics that I talked about in the anatomy of a slime mold. We're going to look at the size and color of the sporangium, although note that many of the sporangium are going to change in color as they progress in maturity. There, you're going to also pay attention to the substrate type. Are they fruiting in wood? Are they fruiting in leaf litter? So those might be very important. And the sporangium itself, it's good to look at the base. If you have your trusty pocket knife with you, you might be able to peel a little bit of the wood close to where the fruiting body is coming out of, so you can see if it has a base and a stalk or it's directly on top of the wood, things like that. And if it's dry, you are going to be looking for those threads through the spores or the capillation. If you are looking under the microscope, then we're going to go a little more specific and we're going to pay attention to the spore size, shape, and the ornamentation. And by ornamentation, I mean, does it have ay Dios mío, warts, or does it have spikes or reticulation, meaning it looks like a little net on top of the spore? All of those details are going to help you make an identification. And remember, the thread-like structures or the capillation can have ornamentation as well. Look at the stalk. Is it just smooth, or are there any structures that you can see? And just keep in mind that most of the genera need the microscopic characters in order to be told to species or brought down to species. Oh, I put here the micro ID is a pain, but the DNA ID is really a pain. I will admit here that when we started the DNA efforts, I really, really, really wanted to do the slime molds. And Megan really, really, really gave me a reality check because in the slime mold world, it's not as simple as do PCR to a gene and do the consensus and call it a day. You have to do repeated cycles of PCR because the DNA that's used to ID it is fragmented. So you have to amplify one site and the other and the other, and then combine them all, and it's a pain. So for now, we will leave that to people with better equipment and software than we do, and we'll concentrate on refulas and bullies. Thank you very much. So now that we have an idea on what it is that we're looking for in the forest floor, let me give you some pointers on who to look for. The first one is the most abundant slime mold in our region. The name is Fuligo septica, but it's also called dog's vomit slime mold or scrambled eggs. And in the graph that you see on your upper right hand is the phenology, or when does Fuliga appear, based on the observations that have been taken on iNaturalist. So these little slime mold is actually not that little. It can be quite big in size, and it can be found even in mulch mounds outside of your favorite fast food. It's that common. It just likes wood chips. It likes to decompose wood. So in the forest, you're gonna find it on top of logs. In outside of industries, you're gonna find it in the mulch. There was a few years ago an article of a woman that was trying to get rid of this slime mold by using a pressure washer or a lot of water from a hose, and hosing it, and the next few days it was everywhere. Well, of course, because what she was doing is giving the perfect conditions for the slime mold to reproduce, and she was being a vector, a disperser of the spores. If you find this slime mold in your garden, don't worry, the slime mold is acting on your favor, is decomposing that wood debris and returning the nutrients to the soil for the benefits of your plant. And if you want to get rid of it, don't wet it. You're gonna make it worse. The second most commonly found slime mold is Lycogola epidendrum. It's also called wolf's milk. But alas, there was, let me not get ahead of myself. So anyway, you're gonna find these in crevices in wood also. It's pretty fun. The peridium of this particular mushroom is a little rubbery. And if you find it in the pink state, I beg of you, press one of the sporangia and see what happens. I really, when I take people out in the forest, I have kiddos and adults alike press on these particular spheres and watch the slime shoot on their direction, which is quite funny for everybody else in the walk. So have fun. Let me know how it goes. So I was very happy to have a slime mold that we can identify as a single species. Yes, it's so easy. Great. Until 2023. 2023 paper came out in the journal Mycologia, and what we thought was a single species was 15 different species that we cannot tell apart just by macroscopic pictures. So if you find these particular group of species in the forest and you want to make the observation, there is now a Lycogala epidendrum complex on iNaturalist. Please select that one. Thank you very much. So there's another one that's very common and quite frankly so beautiful to me, because in my imagination, when I'm looking at Ceratiomyxa fructiculosa, it looks like anemones. So my imagination transports me to the bottom of the sea, even though I am breathing quite fine. But to use your imagination, as shown by this finger right here, you have to really have a hand lens to be able to show the structure of this particular slime mold. Quite fun. You're gonna find this in wood that is a little spongy, is advanced in the state of decomposition, but it's very abundant. And now that we're entering June, you can see by the iNaturalist chart how often you may be able to find this guy. Now, as Valerie was pointing out, there was a little bit of drama with Ceratiomyxa fructiculosa. And for years, I had witnessed people get angry and people get quite passionate about the names of species. And I couldn't understand, like, because of my science background, I was like, but listen, it's just that we're learning new stuff. The names are changing, yes, but it's because the science is advancing. So don't be mad. I don't understand why you're mad. And then somebody dare to say that Ceratiomyxa fructiculosa and Ceratiomyxa porioides were the same species. That happened in 2023. And I, for the first time in the taxonomic knowledge, lost my marbles. I went, how can you tell me such a bananas thing? That is such a bananas thing to say. Don't you have eyes? Don't you see it's so different? Like, this is anemone, this is coral. What are you talking about? So that was 2023, including some club members like, no, they're the same. And I'm like, no. That was the first species that I had a passionate reaction to the change of the name until 2024. Another publication came about and said, no. Ceratiomyxa fructiculosa is anemone, and Ceratiomyxa porioides is coral. They're two different species. And I slept so well after I read that article. In any case, they're gonna be found in similar habitats. It's very damp wood, slightly spongy as well. And typically, there is a little variation to where I find these two. Ceratiomyxa fructiculosa, I will find it more towards the top of the log versus Ceratiomyxa porioides that I'm gonna find inside of the crevices. Now, this is anecdotal on my own experience, but take it with a grain of sand. And then there's a third Ceratiomyxa fructiculosa variety arbuscula. If you ask me, this is just a matter of time before it becomes a third species that is observed in our region. Although this one, it's much rarer than the other two. I've only seen this one myself in the state of Pennsylvania, but apparently, according to iNaturalist, is been seen on this region as well. And I wrote here sus cosmopolitan because supposedly It is found around the globe, but we know, especially based on the case of Lycogola epidendrum, that that may not be the case. Thank you, David. All right. We also have another very fun species, which is the pretzel slime mold. This is much rarer than the Ceratiomixa species that I just talked about. The pretzel slime mold, it's quite tiny. This entire piece of wood, it's about 1 centimeter. But it's quite fun find, and you might be able to find it starting in July. If you look at the graph, there is a peak around the spring. It goes down during the May to June months, but then it picks up again in July, well into the fall months. So check it out. Bring your hand lens, and you might find a friend. And that friend might have a very interesting fungus. So even the slime molds have fungi that feeds on them. And that is the case of Polycephalomyces tomentosus. It is quite rare. It's quite rare to find this little ascomycete fungus that feeds on slime molds, and it appears to be a pathogen of the order, because what you have here is the hemitrichia, and also you have here another trichia species. I'm not quite sure what trichia that was, but this picture is from a past Sequanota, I believe Sequanota 2023. This picture on the left, I stole from the person listed right down here. So you're also going to be finding cribaria, which is a bit rare and can be confused with the reproductive structure of the slime mold Plasmodium. This particular picture was taken with my cell phone last Saturday, and this piece is also 1 centimeter long. This thing is tiny, tiny, tiny. And if you're going to be finding cribaria, you might head up now up to the start of August. Is not a common species, but I've seen it a couple of times already. So head out and look for fallen debris. I've seen these on these cones, but I've also seen it along leaf litter, covering the leaf litter, some of the dead leaves. Then we have the beautiful red Metatrichia vesparium. And that one you can pretty much find throughout the year. May takes a dip, but June and July starts picking up. And I've already started seeing it again this year. To the left is a very lucky picture that I took of Ceratiomixa fructiculosa intertwined with Metatrichia vesparium, which is not something that I see common. So I was very excited to see this. And then to the right, it is a gorgeous picture done by no other than Alison Pollack. You can enjoy Alison's photography in the slime mold identification and appreciation group. She makes fantastic works of art in a picture. And then you also have Hemitrichia decipiens. This was found last Saturday as well. It's commonly known as salmon egg slime mold. It's suspiciously spread around the world, so I have the feeling that this needs to be better resolved, um, like Licogola was. And then is the genus Arcidia or Arcidi. I don't know how to pronounce that. But then you have Arcelia cinerea, Arcelia denudata, and the cotton candy slime mold, I think is named. All of them belong to the same genus. And they have many colors, but they're going to have that elongated sporangium appearance with a stalk that separates them a little bit from the wood substrate that they are growing in. And I love highlighting stemonitis. Stemonitis is another one that's very common, but you need microscopy in order to bring it to species. It's very distinctive as it looks like elongated chocolate tubes. But I also gave you image here of a publication describing one of the species in which they highlighted how much the plasmodium changes in morphology and color from the vegetative stage to the end of the sporangium formation. It's quite beautiful. And Anna, who is one of my nature nerd friends, and it's present tonight, found a stage of stemonitis back 2-3 years ago where you could see all the stages in one, and it looked like, using her words, unicorn barf, but it was so beautiful. So I mean, you could see the whites, the pinks, the light chocolate, and the dark chocolate in the picture, which was fantastic to see. So if it rains, when it rains this summer, head outside and you might see some wonderful works of art that nature has put there for us to enjoy. Oh, we have, let me check one thing. I need to see the battery. Okay. I don't see the battery. Okay, we also have the Tubifera ferruginosa, the raspberry slime mold. It's also picking up now, and it'll be tapering off by the month of September. It's very abundant and it's very distinctive. There's not a lot of things that look like the raspberry slime mold, at least not like Tubifera. There might be a few species, as a matter of fact, they are a few species within the genus, so this is also a species complex. But you're very well seated inside of the Tubifera genus anyway, when you see a morphology that looks like this. Let's see how we are in time. This is how the slime molds have been postulated to be intelligent. And in order to save some of the time, I am going to let you see this at home when you download the presentation. There's a link down here that you can play that video. It's basically, the summary is that they placed oats in the structure of the major cities or the major towns in London and Tokyo. And the slime mold morphology, the plasmodium morphology, was able to replicate how the major arteries and highways had been designed by the engineers. And the way that the slime mold does that is by prioritizing where the cytoplasm and the connections are placed. So when the slime mold first starts going around, it will do so in a radial form, indiscriminatory. But when it finds where the nutrients are, it concentrates the nuclei and the cytoplasm in the shortest distance between the points, so it can maximize the energy where the nutrients are. And that prioritization mimicked how the highways were designed in those two major cosmopolitan cities. And I want to leave you with my favorite resources tonight. Absolutely, I could not tell you enough what a wonderful resource it is. It is having all the experts of every single organism in your pocket is like Facebook for people who love nature. So you are able to take pictures and put them in an account, and then the experts that are active in the community will come and help you curate those observations. So I encourage you to download the application. The iNaturalist application is free, so you can use it anytime. And it is important to note that the better your pictures are, the sharper that they are, the better that we'll be able to help you identify those pictures.
There are
So,
two lectures today.
hello? So the pictures, please make sure that they are sharp, the sharper that you can give them. And please make sure also that you are capturing different angles and different characters that are present in front of you. And then there is a handbook for slime mold identification. Is an oldie but goodie. Myxomycetes by Stephen L. Stephenson and Henry Stemfen. So, what do we do now? Well, join the Mycological Association of Washington, D.C. project on iNaturalist by scanning the QR code at the top. When you scan that code, it should take you directly to the project. And if you already have an account on iNaturalist, you just click join, and all your observations of fungi around the region will be automatically added. And what do you know? Maybe in the next ID table, your pictures will be featured. And then if you're interested in having this presentation, please scan the QR code at the bottom, because I have placed some Easter egg links on some of the pictures and some of the material mentioned, so you can learn more about it. And of course, watch the video about the intelligence of slime molds. So that is all for tonight for my presentation. If you have any questions, I will love to hear them. Thank you very much. I appreciate the love. Feel free to either type on the chat if you have questions, or if you want to use your voice, raise your hand so we can unmute you. You're very welcome. My pleasure. All right. Yes, Mitch, thank you for mentioning Physarum cinereum, gray slime mold on grass. All right, if there are no questions, I will bid all of us good night. Thank you for being here with us to share the energy and to learn all together. I really enjoy spending time with all of you. Take care.